Hello everyone, I am here going to talk about cell cycle. The cell cycle is an ordered series of events. It is the sequence of events by which a cell duplicates its genome and eventually divides into two daughter cells. The cell cycle has two main phases, one is interphase and another is M-phase. Interphase is the time during which the cell is preparing for division. M-phase is the period of actual division. The two timing mechanisms, protein kinase and protein phosphorylation, play an important role in the cell cycle that ensures the orderly passage of all events. This one is the whole process of eukaryotic cell cycle. Before going to the cell cycle, I will say in mitosis, one mother cell will produce two daughter cells. For that, cell have to produce the cellular materials in duplicate manner that is needed for daughter cell. To synthesize the cellular materials in duplicate manner, cell have to go through cell cycle. In cell cycle, cell produce their different cellular materials in different phases. That phases are called G1 phase, S phase, G2 phase and M phase. This G1, S and G2 are called interphase, and this one is the mitotic phase. This cell cycle goes in clockwise manner, not in anti-clockwise manner. M phase is prophase, metaphase, anaphase, telophase. Okay, this G1 phase takes longer period of time, that is 6 to 12 hours, whereas S phase requires 6 to 8 hours, G2 phase requires 3 to 4 hours, M phase requires 1 hour. In G1 phase, cell produce RNA and protein that is needed for S phase. That means cell produce RNA protein in the G1 phase that will be work in the S phase, in the synthesis phase. In synthesis phase, cell will produce DNA as well as RNA and protein. DNA synthesis means they will go through DNA replication, so that two DNA strands will be formed from one DNA strands. That's why this is duplication. That's called duplication of DNA. After that, in G2 phase also they will produce RNA and protein that is needed for the mitosis phase. In M phase, cell division occurs, so that two daughter cells are formed. So the transfer of cellular materials from one phase to second phase will be checked by some checkpoints. There are three checkpoints are there. One is G1S checkpoint, another is G2M checkpoint and M phase checkpoint. G1S checkpoint will check whether the synthesized RNA and protein in G1 phase is ready to go to S phase or not. If is ready, then they will allow the RNA protein to S phase, otherwise they will not allow. Similarly, the G2M checkpoint will check whether RNA, protein and DNA synthesis occurs properly or not. If they have synthesized DNA properly, they will transfer the DNA to the M phase. In M phase checkpoint, cell monitors spindle formation and attachment to the kinetochores. If spindle fiber formation is not done properly, and the cell will not divide into two daughter cells. That's the overall role of eukaryotic cell cycle. The engines that drive the progression from one step of the cell cycle to the next step are a series of protein complexes composed of two subunits. One is cyclin, another is cyclin-dependent kinase. Cyclin is the regulatory component that undergo a cycle of synthesis and degradation in each cell cycle division. Cyclin-dependent kinase is dependent on cyclin. It acts as a protein kinase. There are three major classes of cyclins, each defined by the stage of cell cycle at which they bind CDK and functions as G1 cyclin, S-phase cyclin, and M cyclin. Cyclin bind to CDK molecule and control their ability to phosphorylate appropriate target proteins. This cyclin-dependent kinase also constitute a family of structurally and functionally related proteins that are regulated by cyclin. This cyclin-dependent kinase catalyze the phosphorylation of specific serine and threonine residues of specific target proteins. As different cyclins are present at different phases of cell cycle, the different phases are characterized by the phosphorylation of different target proteins. As different cyclins are present in different phases, different cyclins are there. They are characterized. How they are characterized? They are characterized by the phosphorylation of different target proteins. The phosphorylation events are transient, means that requires a very small period of time, and reversible. When a cyclin-dependent kinase and cyclin, means cyclin-CDK complex, disappear, the phosphorylated substrate proteins are rapidly dephosphorylated by protein phosphatase. Okay. In each cell, if you will see, the single CDK proteins binds all classes of cyclin. Hence, in a cell, there are number of cyclins are there, and only one CDK will bind all classes of cyclin and drives all cell cycle events by changing cyclin partners. Means one CDK will bind to different classes of cyclin and change the events by changing the cyclin. By changing cyclin, they will change the event at different stages of cycle. But in case of vertebrate cell, in contrast, there are more than one CDKs there. So they interact with G1 cyclin, S cyclin, and M cyclin. In mammalian cell, if you will see, at least 16 cyclins and 9 CDK are there. There are major classes of cyclin-CDK complex that control passage through the cell cycle. There are major classes of cyclin-CDK complex are there. One is G1 phase, S phase, and mitotic phase cyclin-CDK complex. Here I am giving the picture where kinase activity of cyclin-CDK complex is there. In G1S checkpoint, cyclin CDK2 complex regulate the process, whereas cyclin CDK2 work their process in G2M checkpoint, whereas cyclin B and CDK1 do their work in mitotic checkpoint. This one is the diagram of animal cell cycle, and we can see here how the activity of cyclin-CDK complex occurs in particular phase of cell cycle. That means this cyclin CDK2 complex will not work in G2M checkpoint or M phase checkpoint. These are specific. Okay. So today we know about the cell cycle and how they process. In the next video, we'll see the regulation of CDKs, whereas as well as all process of cell cycle gradually.